Thank you so much for the introduction and for the invitation to present today. I appreciate all the sponsors in basically allowing this event to go ahead. So my name is Vera Ignjatovic, and so a surname difficult to pronounce, but yeah, don't worry about it. You can just call me Vera. I am coming to you from Melbourne, Australia. I do work for Hopkins, but I actually live in Melbourne, so I have one of these situations where, in the post-COVID world, you can do anything, and yeah, that's where I'm at, and hopefully going to Tampa tomorrow, but with a hurricane happening at the moment, let's see how we go. But anyway, I'll change topic and change lanes a little bit because I'm going to kind of give a little bit more of a broad overview of this healthcare and proteomics and how the two come together. Whether they come together well or not, it still remains to be seen. But let me just change it up. Sorry. Okay. So this is describing some of the kind of elements of my career to date where I've been involved in. About 23 years ago or so, I was involved in starting a hematology research lab for a group of clinicians interested. So this is pediatric hematology, interested in the bleeding and clotting perspective in children, and looking at having a lot of questions, but not really having the laboratory component. So straight out of my PhD, which was actually in Chinese medicine and weight loss, I went to start a hematology lab with no experience in hematology. But it was my actual sports background, which you can look it up if you want on the internet, that gave me this kind of ability to be dedicated and to actually work really hard for something that I want. So I started the lab, and about 6 or 7 years later, I was introduced to proteomics. And the reason that proteomics became very beneficial, was that in the pediatric setting, you are limited with how much blood you can take. And at the time, we were working on healthy norms, and Mike, spoke about population-based norms. In the pediatric setting, which a lot of my talk focuses on, there were no age-specific reference ranges for children, and even to date, a lot of laboratories, when they're doing clinical testing, are utilizing either adult-based reference ranges or published reference ranges for different populations, and pediatric reference ranges are actually lacking. So we're working with perfectly healthy babies born within 24 hours and collecting venous blood samples from them. So we were limited in how much blood we could collect. We were looking at, if we were lucky, 2.5 mL of blood. So this is where proteomics came really useful, in that we could look at a lot more than what we initially planned to. And proteomics then allowed me to expand, looking at only pediatric thrombosis and hemostasis to looking at a variety of different clinical scenarios. And some of them are mentioned here. So I've worked with clinicians. I'm a biochemist. I've worked with clinicians across many, many different settings. And I've also been involved in the marketing committee of HUPO and so on. This is some of our kind of more recent work in the proteomics space. And so, like I said, it's not the only thing that I do, but it's super, super useful, especially when looking at some of these clinical scenarios. Now, we've spoken about innovations and advances in healthcare. This is the daily reality for children in the ICU that are placed on heart-lung machines. So this is extracorporeal membrane oxygenation, essentially a heart-lung machine that replaces the function of the heart and the lungs for a certain period of time, allowing these children to get better. You can see that the blood is running through hoses. It's a hose. It's essentially like a garden hose with many, many different connectors and very long length. And we know that when blood comes into contact with the surface, it starts to clot immediately. So we have this scenario. Like I said, it's very archaic. This ECMO scenario has not changed basically since the start. Nothing really has changed. And what we have is increasing number of children going onto ECMO, and the outcomes, whilst for some children are really good, we have about 70% of these children that get a blood clot in the circuit. And also, how is a blood clot detected? So this is current medical practice. Nurses are coming with their phones and the actual flashlight turned on, going down the circuit, trying to locate a blood clot. This is happening, basically in this hospital, pediatric hospital, many hospitals worldwide. So we don't even have a way of detecting these blood clots. They block the circuit needs to be changed. Of course, this is, the patient, then there is a hard stop essentially on their care for the circuit to be replaced. Every circuit costs about $50,000. This treatment is really expensive. So we have clotting in the circuit, we have bleeding in the patients, and we have clotting and bleeding in some patients. So it's quite complex. What is done is, in order to prevent blood from clotting, is that anticoagulants are given to these children. So heparin is used in a lot of these circuits, and some newer drugs are also, bivalirudin is also being used lately. But what happens is that a lot of people have studied the blood, but nobody has really looked at the circuits until we started doing it very, very recently. So the circuit before the patient comes into ECMO is primed usually with albumin, with an albumin solution, so that the albumin attaches, this is theoretically attaches to the circuit and prevents other proteins from binding, which is fascinating because we know this is the concept of the albinome. We know that hundreds of proteins bind albumin. Why this is even happening is puzzling, but that's the best care available to these children, and also adults are placed on ECMO very frequently at the moment. So what we did recently was we actually collected the circuit that was primed before the patient was attached to the circuit. So this circuit was not, and it didn't end up being used. And we looked at what is binding to the circuit once it's primed. So we have about 120 proteins that are bound to the ECMO circuit before the circuit is even applied to the patient and before it's being used. And then what we also did was we actually looked at circuits that are removed from patients and are normally discarded, normally thrown into the rubbish bin, and we did proteomics with this. So this is DI-based approach, and I know that Neil doesn't necessarily agree with this, but I love your proteoform work, and it's super, super important in the pediatric setting, super important, and we can talk about that later. But we can see that there are 933 proteins that are binding to the circuit that are common to every patient. Now, these are 6 patients, different ages, different length of, different duration of ECMO, and different outcomes. But we can see that, not what we are measuring in blood, or what the clinicians are measuring in blood, to actually see the effect of the drugs that are given to the patient. What they are measuring is actually what they can see. They're not measuring what they can't see. And a lot of these proteins are attaching to the circuit. They're not in circulation. But if you can imagine that drugs are given to target certain proteins, well, what if they're not in circulation? What if they're being attached to the circuit? So this is where I think proteomics is super, super useful. And then what I wanted to touch on, in this fit for purpose talk is when you see this hierarchical clustering plot, a lot of the proteins here are missing. Now, a lot of people in the proteomics field have a problem with missing proteins. They love to fill in these missing values with numbers. Now, in this clinical scenario, if we did that, we would actually be completely covering the differences between these patients, which is what we don't want to do. There are proteins that are bound to the circuit, and there are proteins that are not bound to the circuit, and this is different between patients. So this is where it's very important, if you're thinking for fit for purpose, to talk to the bioinformaticians and to say, no. We don't want to fill in the missing values. We need to work with this data as is. So this is, I think, another element that we need to really think about when we're thinking about being fit for purpose, or proteomics data being fit for purpose. Because a lot of people click the button, fill in missing values, and off we go, which I think that we need to think about carefully whether that is appropriate for what the scenario that we're looking at. And clearly in this scenario, if we're looking at what's bound to the circuit, we do expect missing values, and it's completely okay, and we need to work with that. So if we're thinking about this translational research scenario, from discoveries to then the community side of things, and it's a continual spectrum. I don't see it as, one versus another. we need to really think about, whether what we are working on is suitable for the use for which it is intended. So keeping in mind that, it is actually compatible with existing patient care. Yes, we want to look at the future. Yes, we want to look at the big picture. Yes, we want to innovate everywhere. But if we have a scenario that hasn't been innovated and it doesn't look like it's going to be innovated in a long time because it's working as intended, saving lives, questionable to which degree, but it is. Nothing has really changed. We can't say, oh, we want to work, think about what ECMO looks like in 20 years' time and set up our studies to match that. We need to be able to set up our studies to match what is currently happening. And if you think about the innovation perspective, many of the presentations today have focused on microsampling, and I think microsampling is super important. So a lot of innovation has happened from the sampling perspective, from the processing perspective. Yes, everything is quicker. Yes, data, we're getting a lot of data, and data is being processed quicker. So all of that is wonderful. And again, all kinds of different approaches to microsampling. This is where, this is where we are at and where the future of healthcare is. And I think some aspects of healthcare are already there. Facial recognition technology to ensure the sample is being collected from the right individual and so on. Shipping in temperature and humidity controlled conditions, all of that is great. And we, have the capability, why not, to have these samples delivered by a drone so that it's directly from the patient direct to the laboratory. And I think a lot of that is probably already happening in some scenarios. We also have this technology that allows at-home monitoring of whole blood, things like the whole blood counts and so on. And this particular one is for treatment of schizophrenia, and it's associated with neutropenia. So you can monitor at home how the treatment is going and whether neutropenia is, whether it's getting worse or not. And the patient doesn't even need to leave their home. So they put the drop of blood on the cartridge, insert it into this analyzer, the results go straight to the clinician. So everything is fine. All of that is great. We have in the healthcare ecosystem many, many advances shown here on the left side. So we have surge in healthcare spend and investment. We have scientific investments, like I said, a lot of innovation in the digital and AI space. And also we have alternative healthcare models, which is hospital at home and so on. But we do really need to think about that in the context of what's shown here on the right side, which is this widening global health disparities, the healthcare worker shortages and their burnout. It is a problem, not just in the United States, in Australia. It's a problem in Australia, in Europe, and I think in many, many countries. The worsening mental health and well-being, not just from the adult perspective, but it's seen in children as well, and as well as these kind of macroeconomic issues, such as the inflation, the climate, and energy crisis, and all kinds of political problems. And I think that then we really need to balance this out. And when we're thinking about these proteomic innovations versus the current constraints in healthcare, and also thinking about this idea of the research bubble. So what do I mean by that? Many researchers focus a lot on when are the samples collected, the timing of the samples, what kind of temperature is the sample kept at, how is the sample processed, how is it spun, all of these kind of things, how is it aliquoted, everything, how is it then stored? So a lot of thought is being placed into that. And many studies, months and months are spent for people to standardize all of these kind of all of these factors. But then what happens is on the clinical side, the sample is collected when the person is available to collect it. It's not collected, yes, the aim is to collect it at a certain time, but frequently it's not. When the person is available, that's when the sample is collected. collected in a whole lot of variety of tubes. And we focus so much, I think, in the proteomics space on we need this sample type, we need this sample type. Well, I think we need the sample types that are used clinically. And, this is only just a small kind of representation of the different types of tubes that are used, heparin tubes, EDTA, citrate, what are we actually collecting the sample in? And I think that trying to match the clinical scenario as much as possible is probably important. And then what happens once the sample is collected? We think about it in a research scenario. Okay, we need to carefully take this sample around and process it and so on. Well, guess what? In the clinical scenario, it's placed in a pneumatic tube. And what happens once it's placed in a pneumatic tube? This is how it travels. This is how it travels to the lab. It's being thrown around, basically on the back end, being thrown around, arrives to the clinical lab. And what happens in the clinical lab? Well, if the clinical lab is really super advanced, someone will unscrew that pneumatic tube and place the sample on an automated track where the sample is then processed in an automated and controlled fashion. Phenomenal. But it's not like that in every lab. In many, many labs, the sample arrives at the lab. It's processed when the person is available to process it. It's placed in the centrifuge. The centrifuge is started, and I've seen it with my own eyes. People forget. People remember that sample is in the centrifuge. When the next person comes to centrifuge the next sample, they're, oh, okay. So this is what happens clinically. So I think just being aware that, we can create a research bubble, but then our findings really relevant to the clinical practice where things are done very, very differently. So this interface, I think, between research and the clinical practice and people being cognizant of both are really, really important. And that's where I think that it's really important that we give a chance to our PhD candidates in particular to experience that whole spectrum and to allow them to go to the clinic and see what the clinician is doing. So if the project involves a collaboration with a clinician, to allow the PhD candidate to spend a little bit of time with a clinician to see how the samples are collected, to go and see what happens in the actual lab, so that their project can then be set up to actually match the clinical practice rather than some kind of an idea of idealized kind of conditions. Now, I hear a lot of talk about mass spectrometry being a black box and it's so far away from clinical practice. But I think what we forget is that it's already in the clinic. I mean, the huge advantage of proteomics is that, hey, we have mass spectrometers in the clinic already. We need to leverage the fact that they are there already. So that kind of barrier is already overcome. So we have this equipment already in the clinical lab, which is a huge plus, so that we don't have to really reinvent the wheel from that perspective. But what we have to think about is these kind of issues. So sample type. Very, very frequently, I hear people talking about plasma and serum interchangeably, very frequently. And they're not the same sample. Also, what we have to think about is we talk about innovation, but the whole space of inflammation uses serum. Guess what? When you are creating serum, you're activating the coagulation cascade. The coagulation cascade is pretty much overlapping with the inflammatory system. They don't operate independently. So when you're activating the coagulation cascade, you're activating the inflammatory system, you're increasing these cytokines, and then you're measuring them in serum. It just does not make sense. But the whole world of, every inflammatory markers are measured in serum, standard practice, no problem at all. It actually does not make sense. So I think we need to go back and rethink some of these things. And also people say, oh, I don't want to work with plasma because of high abundance proteins, or I want to remove them. I love the fact that Jessica mentioned that albumin is important. I think it's super important. I think these high abundance proteins, and I saw, Mike, in your talk, some of these alpha-2 microglobulin, I think it was some of these super high abundance proteins, they are important. They've been conserved across species, and they've been conserved, and they are there for a reason. So I think that people that say, oh, I want to look deep into the plasma protein, fine, phenomenal. But if you can detect, if you can get an association between a high abundance protein and a clinical outcome, it's actually easier to translate it to the clinic because it's easier to measure it in the clinic using a quicker protocol. So I think we have to think about in the proteomic space of, high abundance proteins are not bad. Let's not give them this kind of bad guy tag. Then the way in which we detect and quantify proteins, I think it's really important. I think that the fact that we have a variety of different technologies is wonderful. And I think that we need to utilize all of them and see which one works best. Because if you think about it, in current clinical care, at least when we're thinking about the thrombosis side of things, the bleeding and thrombosis side of things, the clinically useful assays, and I'll mention some, there is PT, APTT, fibrinogen, so on. These assays have nothing to do with physiology, nothing at all. They are completely irrelevant when it comes to physiology. So the way in which a sample is activated, the way in which this clot formation is measured, nothing to do with physiology. But the good thing is that there is a number associated with an outcome. And that's what it's important. As long as there's a number associated with an outcome. So it doesn't matter what protein it is, as long as that protein or the particular proteoform or particular protein level, concentration, and so on is associated with an outcome. It doesn't matter. It doesn't have to explain biology as long as it's associated with an outcome. And that's, again, where I think we spend a lot of time. We need to explain how this protein is associated with biology. I think we're just wasting our time. We don't need to. If it's associated with an outcome, if it can be used to predict an outcome, that's good enough. And also, again, think about this, targeted versus untargeted approach. There is a value for both, I think, and also limits of detection. So are we detecting what is present versus, looking for what is absent? And on this translational discovery pathway, it's really important to focus on clinically relevant questions. So I think that, yes, advancements in the technological aspects are super important, but I think for people that are working on that intersection between, research, so clinical, that are working on the intersection between clinic and research side, laboratory research specifically, we need to think about, are we working on clinically relevant questions? So that's where, again, I think the opportunity to work with clinicians and to actually interact and to say, okay, how is this going to help your population? And even with the clinical researchers, they're thinking about how to analyze their data. And it's like, well, what is the question that you would like to know? What is the answer that you would like to know for your population? Let's set the study up to actually be able to answer that question. So collaboration between laboratory scientists and clinicians, I think, is super important, and to actually speed up this translation of findings to patient care. So it's basically going from nothing to something, really. And how do we do that? So I wanted to spend a little bit of time speaking about the model that we have at the Johns Hopkins campus, but this is the campus in St. Petersburg, Florida, where they're currently experiencing a hurricane. I think hopefully it will pass by, but let's see what happens. So we established a laboratory that we're calling the Translational Research and Innovation Laboratory. And this is a very different approach to the traditional laboratories. And I think this is a lot of what we are thinking about on the research side has focused in the past, or is focusing even to date on this traditional model of a laboratory, where someone goes to do their postdoc, to learn a particular method, to become their independent researcher and so on. But with such massive advancements in technology, every lab can do everything. We like to think that, you need to be a mass spectrometry expert, you need to be an expert. No, you don't. I think there's a lot of opportunity for breadth. Yes, you do need people that are experts in certain things, but being able to cover a wide breadth is really, really important. And very specialized laboratories are becoming very, very quickly a thing of the past. And very specialized postdocs are also, I think, becoming a thing of the past. And hopefully as senior people in this room senior researchers are thinking about how can we provide this really wide experience to our PhD students and to our postdocs, so that they can actually be able to ask the right questions, put the data together, rather than be really specialized in a single technology. So in our trail laboratory, we really focused on bridging the gap between patients and laboratory science through teamwork and innovation. And we are looking at many, many different aspects. So we are looking at hemostasis. That's my kind of initial focus. We do microbiology testing. We do panomics. We do focusing on microsampling and trying to change some of the ways in which samples are collected. Particularly thinking, microsampling is super useful when thinking about individuals or families in the pediatric scenario coming from low socioeconomic status. I think that this argument of coming to the hospital to get a sample, I think from an adult perspective, it might be a little bit different. But if you're thinking about it from a pediatric perspective, where a parent has to take a day off, the child has to take a day off from work, child has to take a day off from school, they have to go to the hospital, they have to find parking. In many hospitals, they have to even pay for parking. All of these things to get a sample. And I think being able to do microsampling at home is very advantageous. We have collected samples, finger prick samples from children in the emergency departments, where we're looking at concussion and delayed recovery from concussion. And I can tell you the only problem with microsampling or giving blood samples is with teenage boys. They are scared of giving blood. Everybody else is super, super fine. So we have teenage boys, I think, afraid of fainting in front of everyone, that perhaps a little bit wary of giving blood. But yeah, I think thinking about microsampling is super important when it comes to socioeconomic status and those considerations. We are also looking at assay innovation. We mentor clinicians and we mentor scientists that are doing research and provide consulting on how to set up the projects. And this is in the context of Johns Hopkins took over a private hospital approximately 10 years ago, a private hospital with no academic mission. So we're building an academic mission from scratch. So I think having a laboratory that's not specialized is super important, because we can then be able to help everyone, to help clinicians that are coming from very different spaces. And we use a hub and spoke model. So we have a super, super small footprint. But what we do is we leverage our network where we are connecting via service agreements, collaborative agreements, and so on, with different facilities. So we use one facility. We use Jennifer Van Eyck's facility at Cedars-Sinai when we want to do DIA. We use a facility in Baltimore where we want to do TMT. We utilize Vanderbilt University when we want to do Olink. They have a facility there. We utilize a laboratory in Nottingham, England, where we want to look at quorum sensing molecules for our microbiology work. So this model is working super, super well for us. And it allows us to look at different things, looking at albuterol concentration in urine. We look at red tide exposure, looking at brevetoxin, and particularly that's really important for the Tampa Bay area. Because a lot of people fish, especially in the low socioeconomic status. A lot of people rely on the fish that they catch, which is free, and eating the fish from the Tampa Bay area where there are a lot of pollutants. And we're also looking at recurrent thrombosis, so looking at children that have a blood clot and trying to predict which of those children are going to have a recurrent blood clot. And I can tell you everything that I showed you about the Trail Lab. We have one person in the lab. One person in the lab, but Stephen is our gold. And when I came into my position, I was really focusing on thrombosis and hemostasis, but being able to leverage this kind of experience is phenomenal. So this is, again, an argument towards really not kind of getting people to really deep dive and focus on one thing. So Stephen has experience from a clinical perspective, where he's worked at a molecular genetics lab at the Johns Hopkins All Children's Hospital, looked at NGS panel development and so on. He has industry experience as well, which is excellent, and then research experience as well. When he's done some DNA fingerprinting for algae species and identification and so on. So being able to recognize, okay, we have an individual that has such a breadth of experience, and saying, wow, this is excellent. Because if you think about it, all of our funding streams are really still focused on individuals having that kind of a deep dive, whereas the world that we're working in is very broad, and things are changing very rapidly. So Stephen doesn't have a PhD. It doesn't matter. It doesn't really matter. But he can pretty much do it all. And we have this broad group of clinicians. So we have Dr. Neil Goldenberg, who is co-leading the lab with me. He's a hematologist. We have Marisol Betansky here. She's a hematologist. We have Dr. John Morrison. He's a hospitalist. Tony Soche, he's a critical care medicine fellow. Amy Kiskedan, PharmD. We have Maha, who is hematologist as well. And we have Jen Lighting, who is an immunologist. So we have this wide group of clinicians. We meet weekly. We discuss any given topic in front of everyone. So we have this variety of opinion. When we're discussing microbiology essays for Dr. John Morrison, everybody's there. Everybody's asking questions. Everybody's contributing to the discussion. And I think that's really important. And then when it comes to consulting, we're working with clinicians to plan their projects. How do we, from anything from implementation of the project to looking at feasibility and budgeting, we do that up front. We think about which biospecimens do we need to collect, and how do we use them. And then we connect with existing capabilities and expertise. We have an automated CAP-accredited biobank, which is huge investment for that campus, and we're utilizing that really, really well. We have existing cohorts which are biobanked, so pediatric cohorts, and we try and use existing samples wherever possible to try and generate the preliminary data to be able to do larger studies, larger funded studies. And we do some proteomics. So we do think about experimental design and consultation. What is that sample that really needs to be collected? And we worked with a variety of samples, including breath condensate, which is super fun to work with. Like I said, we collaborate with external laboratories and facilities. And what I really wanted to speak about is, yes, proteomics data is complex, but it doesn't need to be. So we utilize, and free plug for Mass Dynamics platform. I'm an Aussie. I've lived in Australia for a long time, even though my accent doesn't sound really Aussie. But a huge plug for Mass Dynamics. We are utilizing the Mass Dynamics platform where clinicians are interacting with their own data. So they're being able to interrogate their data to ask different questions. They're doing it on their own, and it's super easy for them. We don't always have to go back to the source lab to ask yet another question, yet another figure created, change the colors, do all these things. So we can do all of that with the Mass Dynamics platform. And it's been super useful for us, and it's made the proteomics data super simple to understand for the clinicians, which is where if you want to get a buy-in from the clinicians, not speaking their language doesn't really help. They will just walk out of the room. But being able to actually speak their language and being able to allow them to interpret the complex proteomics data in a very simple fashion is really, really useful. So I want to show two examples of the kind of things that we're doing. So in this particular scenario, we're looking at children with tracheostomies. And children that get tracheostomies, and even adults, one of the kind of bad outcomes is a tracheostomy-associated respiratory infection, or TARI. And that infection is associated with frequent hospital readmissions. So the patients are sent home. They are able to live with their tracheostomy and lead a relatively normal life under the circumstance. But they frequently have to come back to the hospital because they're getting these infections. So what we've done in this particular scenario is we just wanted to kind of explore, if we collect plasma, urine, and tracheoaspirate samples, and we do the Olink, in this case, the Olink targets 48 cytokine panel, and again, Mass Dynamics for interpretation. How, what do we actually see when we look at these specimens? Do we need to focus on one versus another? Should we work with all three, and so on? And this is, sorry for the very busy plot, but I just wanted to show you here in red On the top left, in red is the plasma, and this is how the plasma samples are grouping very differently to the tracheal aspirate samples here in green and the urine samples here in blue. And I just have to say that plasma samples are the easiest to collect. I mean, blood is super easy to collect. Urine is not easy to collect. Maybe from adults it is, but we are working in the pediatric scenario. It's not really easy to collect from children. And also tracheal aspirate is difficult. Whilst it's collected clinically, it's difficult to work with because it's kind of this mucus, really kind of not a very homogeneous sample, and a lot of it is collected in the presence of saline and in the presence of different volumes of saline. And then we have to think about, okay, how do we normalize what we're actually detecting and so on. And we can see proteins that are differentially expressed between the samples and so on. And we can see which proteins are kind of correlating nicely between the samples and which ones are not. We haven't really focused we haven't done a deep dive on this particular data because we know that, okay, we can get a signal with all three specimens. We're trying to collect all three if we can, because we know that if we do the discovery part of the study in one particular sample, we can then look for those markers in a different sample and see whether we can detect them and see whether, for example, in this scenario, whether the tracheal aspirate samples are actually, even though they're more difficult to kind of work with on the proteomics side, whether they're clinically easier to collect. You know, we have to think about these things, and we have to think about what is easier for clinicians, what is easier for the nurses that are there collecting the samples, and which sample type would they actually want to work with the most. And then another scenario is looking at children in the ICU. They get acute respiratory failure. So they get invasive mechanical ventilation, similar to ECMO that I showed you earlier. And complications are associated with, with a longer length of IMV duration, so with longer length of invasive mechanical ventilation, and also with increased oxygenation index. In this case, we ran TMT proteomics at the Baltimore site, and again, looked at data interpretation with mass dynamics. So here on the top left, we were looking at change in the plasma proteome based on the duration of invasive, or I would call it IMV. It's difficult for me to say it. So we can see that most of the individuals where the length of IMV is less than 4 days are actually grouping together, and they are associated. We know that they're associated with better outcomes. And then we can look at markers that are predictive of that length of IMV duration, because the bottom line here is not to actually see what are the differences, but to actually be able to see which are the proteins that at the start of invasive mechanical ventilation can predict which children are going to have that delayed invasive mechanical ventilation, and which children then might require different clinical care. And here on the top right, we looked at changes based on the oxygenation index and increased versus high versus low oxygenation index, and very similar to the scenario on the left. We were looking at, in this case, what are the proteins that are differentially abundant when it comes to these two different scenarios. But again, thinking about the baseline samples and how can we predict, or can we predict, which children are going to have that increased oxygenation index, and then a more complex outcome, and might require different care. And something that hasn't been really mentioned today, and I just wanted to mention at the end of my talk, is this concept of decentralized biobanking. I think as researchers, we really haven't been accountable for the samples that we are collecting. I think we haven't. And the reason that I'm saying that is that on average, in any automated biobank or in any biobank, about 17% of the samples are utilized. So people collect a sample, they aliquot it out into 6, 7 aliquots, 2, 3, it doesn't matter, but only 1 aliquot or 2 are used. And the rest is just remains there. The researcher moves on to something else. The samples remain stored and are not used at all. So this concept of the decentralized biobanking is really changing things on the patient involvement in research side and will change accountability for researchers, because we need to engage individuals in biospecimen research. We need to empower patients as owners of their samples, because I think that this argument that we've had for a long, long time of, you are contributing to somebody else's, improved outcomes, I think it might not fly. I think that my patients want to see, hey, can you in real time improve my care? Is this going to have a change for my actual stay in the hospital? Or yes, okay, fine, that it's going to help someone else, but how is this going to improve my length of stay in the hospital for this particular episode, for example? Connecting with samples and results, I think, is really, really important, because, we all promise, or at least in Australia, there's a promise that, research findings will be sent to the patients. I'd be interested in how many studies actually at the completion of the study and at the end when the findings are published, how many people go back and say, oh, let's do a summary of our findings and we'll send it back to all of our participants and make sure that they're informed. I don't think that many people do that. And then, thinking about this tracking and learning about the specimens that are collected and stored for research, thinking about it from a perspective of how can we utilize samples that are collected, for example, in one institution, how can another institution utilize them? Also, how can we monetize? How can we create a financial reward for the participants? So how can they be the owners of their samples and be able to leverage their samples to empower their understanding of their disease scenario? So for example, in rare disease scenarios, laboratories are working with very small sample numbers. And the argument is, yes, it's a rare disease, small sample numbers. But what about if decentralized biobanks are used so that the patients with a rare disease can actually empower the research field by saying, okay, we'll unite all of our samples. So now you're not looking at, yes, it's a rare disease, but now you're looking at increased numbers of samples that you can work with. And also knowing what has been done with particular samples. So not running the same cytokine panel on the sample on which it has already been run, something as simple as that. So decreasing the cost again and increasing the efficiency in being able to use the data that already exists. Who ran the test, who has this data, can we get access to this data and build on it rather than kind of start again from the same spot. And again, like I said, this will help to enable collaboration and to enhance learning and really to accelerate this precision medicine concept as an equitable standard of care. So I think that's something that we really need to think about, and we'll be forced to think about it from the perspective of, is the research that we are doing fit for purpose, fit for that patient, fit for that patient population, and so on. And I'd like to end my talk by saying, if you want something new, you have to really stop doing something old. So I think that this, the way in which things are done, just because everybody's done it that way, I think we need to kind of really quickly stop with that. And we need to think about, okay, it's not about which sample do I want collected, but it's about which sample is collected in the clinic and how can I leverage that sample to actually add to what is being done rather than kind of reinvent, create a solution that is never going to really be useful for clinical practice because it looks very different to what I kind of imagine it. So yeah, once again, thank you for the invitation to present to you today, and I'll be happy to answer any questions. Thank you.